you might be wondering why we're looking at pasture-based dairies. There is a group of scientists from across Utah State that are working on this. And we're looking at ways to try and improve those dairies because we have so many milk cows in the Western United States, and we're starting to see a huge increase in the number of pasture-based and organic dairies. That demand for organic milk has increased steadily, and we want to help find ways to improve that. Those organic dairies in particular are required to graze for 120 days in a year, with a minimum of 30% of that dry matter intake being required to come from a pasture. Now, that's all seems great and fine and no problems, but when you actually start to look deeper, oftentimes we're seeing reductions in the dry matter intake when we move livestock onto pasture. And in the case of milk cows, we're often seeing up to a 32% decrease in the milk production. That's a big hit. And so we're trying to find ways to help make those organic dairies and anyone wanting to do grazing improve their yields and their productivity, and trying to find ways to reduce that impact. Organic dairies in particular, we often see a lower herbage productions. That's partially just because the organic fertilizers are more expensive. And so even if you're just going to a point of economical return, you're not going to put as much nitrogen fertilizer on because of the cost of the fertilizer. And I think in many cases, they try to, because that fertilizer is expensive, they try and cut that down even more. But we're also seeing in many cases lower herbage production or mass production. So our goal was really to try and help identify highly productive, economically and environmentally sustainable grazing systems. We're looking at ways to maximize the rates of gain, maximize that forage production, again, to a point of economical return, and we want to protect the environment. And in particular, in a grazing system, we're looking at protecting the groundwater. Now, you might be wondering why we're concerned about the groundwater and the environment in a pasture setting. I mean, it seems almost like your perfect little Norman Rockwell setting, beautiful little livestock out grazing on a green pasture. What could be more beautiful? And they're consuming nutrients off the pasture and recycling them back onto the pasture, minus what few they keep as they grow or produce milk. But when we look further, it's not quite so benign. The distribution of the nutrients is not uniform across that pasture. So they're pulling nutrients off from across the whole pasture. And in a given year, only 10% of that pasture will receive any nutrients back in the form of a urine or a fecal spot. Urine especially is problematic. Urine can contain up to 1,000 pounds of nitrogen per acre in that urine spot, just that little tiny spot. But that's way, way, way more than our plants can possibly use. And so most of that's either leaching, going down into our groundwater, or volatilizing. The other reason we really want to look at the environmental part of it is that we know that AFOs and CAFOs, so operations that have livestock in confinement for 90 days or more out of the year, have to meet certain regulations. EPA has dictated that. They have also proposed and tried to push through regulations for grazing operations. Currently, those grazing operations are exempt, and they kind of backed off after they first proposed it. But I would fully expect that that will come back to bite us, that we will have regulations on our grazing operations. And all we really have to do is look at Europe, and they also regulate a lot of the grazing operations. And that's because they do realize that there can be negative consequences to it. So we actually started on a long series of studies. We started way back with a study looking at beef steers when we wanted to see if we could add legumes into the system to reduce or offset the cost of nitrogen fertilizer. In this study, we were actually even using inorganic fertilizer, but fertilizer prices were skyrocketing at the time. And so we thought, well, if we add in a legume, maybe we don't have to put in those fertilizer, that buy it so much fertilizer. We used tall fescue, a very highly productive forage grass, although not usually the livestock's favorite. And we added in alfalfa, bird's foot trefoil, and then we had one with tall fescue with regular inorganic fertilizer, and then tall fescue with no fertilizer. As you can see, tall fescue with no fertilizer does not do well. The one that actually did the best was our tall fescue with bird's foot trefoil. And when we actually looked at it in further detail and took into consideration the costs for the establishment and maintenance and the stocking density, bird's foot trefoil really shone. It had the highest stocking density. It had one of the lowest costs per acre for establishment, and it had the highest return per acre, net return per acre. And so we found that bird's foot trefoil really was a good option for us. We still thought we should be able to improve on those rates of gain. And so we looked at them, and both our alfalfa and bird's foot trefoil ones had a higher energy level. We wanted to know if we could possibly improve that. And so we actually, in this study, we looked at putting in a high energy grass. So we used a high carbohydrate perennial rye and a high carbohydrate orchard grass. And we're looking at them with and without bird's foot trefoil. So here's our study objectives that I'll report on for this study. And we're again trying to improve our forage yield. We want to reduce our fertilizer costs and improve the economics. And we're doing that. We're also looking then at our livestock gains. That's the bottom line. That's the important part, the livestock gains. And we're looking at the environmental impacts. And we also want to look at the reproductive impacts. As we add legumes into the system, we're adding nitrogen into their diet. We wanted to make sure that we were not adding too much nitrogen into the diet and causing any delays or reproductive development issues. So here's our materials and methods. We had 8 treatments. We have 4 grasses: tall fescue, meadow brome grass, a high sugared orchard grass, a high sugared perennial rye, and then those 4 grasses with bird's foot trefoil. And we also had a control total mixed ration diet. And those were kept out at the cane dairy at a separate location, but then brought in for weigh days. For those pastures, we used Chilean nitrate. And this isn't, so we were using organic production methods for this study. We also added in feather meal. The grass monocultures got 2 doses of Chilean nitrate. We gave the grass legume mixtures 1 dose of Chilean nitrate early in the spring, just to help get the grass started and kicked off. We irrigated every 2 weeks throughout the summer. And we had, for this study, we actually had, for the rates of gain and things, we had 3 9-acre pastures that we were looking at. For the environmental component, that's only on 1 of those pastures. So we have, and that's this picture here, the little white thing in the corner, that's my trailer, our command central for it. But we have our 8 treatments. We had Jersey heifers on each treatment. We were using a rotational grazing system where they would graze for 7 days, then be moved to the next paddock and graze for 7 days. Took 5 weeks to get through that. We would take them up to the main facilities, weigh them, collect urine and fecal samples, blood samples, check on them, make sure that everything's going well, and then put them back out and start that grazing cycle over again. We ran through 3 grazing cycles per year. We do try to make sure that we have the livestock off by September so that we can let that bird's foot trefoil rest. If you've heard that bird's foot trefoil is really fussy and doesn't hold up well, we're finding it does really well here in our environment, but we also are making sure that we let it rest and build up the root reserves in September. For our fecal samples, we collected those the fun way, grab samples. And so it's really fun doing all of our little heifers with a grab sample. Those samples then were analyzed by for total nitrogen using a combustion method. The urine samples, another fun process. We get to tickle them enough till they pee, and then we collect that pee and we analyze that for urea on a latchet flow injection analyzer. And under Dr. Kara Thornton, they were collecting blood samples and they analyzed those for blood urea nitrogen. Out in the field, looking at just how those nutrients actually were moving through the full system, we have both zero tension lysimeters where we collect the leachate biweekly. The zero tension lysimeters are actually big soil cores, 15-inch by 44-inch deep soil cores that were extracted. A collection system was put on it and they were put back into the ground where they came from. We then removed that top portion after the soils around it of that PVC pipe. We can actually plant and till over these zero tension lysimeters. They're really, really sweet, but they're expensive and difficult to put in. The leachate from the zero tension lysimeters, we're analyzing for nitrate nitrogen on the latchet. And we also have suction cup lysimeters out there. These we collect leachate every week through the growing season. So from as early as we can get out there in the spring till as late as we can get out there in the fall. And those are collected weekly. You put a vacuum on, it's a little PVC tube with a little ceramic cap down at the bottom. We have 3-foot tubes so that we're pretty well past the root zone for this study. And then we collect that leachate and take it back and analyze it for nitrate nitrogen. So now to the results and the fun stuff. What were we finding on the average daily gains? We found that in all cases, our mixtures, our grass legume mixtures with the bird's foot trefoil outperformed the monoculture. And we were perennial ryegrass, the average daily gains were right up there with the TMR. So we're doing really quite well in this study on that. When we look at the urea concentration, so the urine, we are noticing that we're seeing a lot more nitrogen in those mixtures, even more than, and some of them than the feedlot diet for the perennial rye. That's not necessarily surprising. We're adding in a legume that has a high protein content, and that protein is actually digested and broken down in the rumen very easily. And most of that protein that's broken down in the rumen will go into the urine. The bird's foot trefoil helps some because it does have tannins, and those tannins bind to those proteins in the rumen and then aren't releasing them until they get to the abomasum, which gives the animal a better chance of being able to absorb and utilize more of those nutrients. But seeing an increase in that nitrogen content isn't surprising. What we're concerned about then is how much of that is increasing the nitrogen out in the field that's getting down to the groundwater. And when we look at the fecal nitrogen, we're also seeing an increase in our mixtures, not quite as much. Having an increase in nitrogen in the feces is actually really not a huge problem because most of that nitrogen is in an organic form, so it's tied up and it's released slowly. So that gives the plants a chance to be able to utilize it. It's the urine that's really the problematic one. And you'll note that these were actually lower than most of the feedlot ones for the fecal nitrogen. The blood urea nitrogen also tended to be higher in the mixtures than the monocultures. Again, not surprising. You add in more protein into that diet, more nitrogen, and you're going to expect to see more nitrogen in the urine and the feces and the blood. The reason we want to monitor that blood urea nitrogen is then for the reproductive aspects, and Dr. Clay Isom is looking at those, the reproduction aspects. So the leaching side, what were we seeing? On the nitrate for the suction cup lysimeters, we found that there's actually a huge difference based on grass species. In this study, perennial ryegrass by far let the most nitrogen go through, especially in the monoculture, and yet it dropped tremendously when we added in a legume. And the orchard grass was second, and it also dropped tremendously. Meadow brome and tall fescue, both the monocultures were slightly lower than the mixtures. They also, in general, did a much better job of capturing those nutrients. My own guess is that's really, a lot of that has to do with the rooting system. Orchard grass tends to have most of the roots in the top few inches. And so the bird's foot trefoil probably can help fill in better down below and catch more of those nutrients as it's moving down past the majority of the orchard grass roots. for nitrogen on the zero tension lysimeters, the results are not exactly the same. You know, you would think they should be, but that's probably not totally unexpected. We do get some spikes as we get urine spots that hit those different lysimeters. And so that, depending on exactly how many urine spots you get, that can kind of tweak things a little bit. Again, our perennial rye was the one that exhibited the most nitrate loss. Orchard grass was next with meadow brome right there. And in this case, we have three of them, the orchard grass, the perennial rye, and the tall fescue, that all actually exhibited slightly lower levels of nitrate leaching in the mixtures. And phosphorus, I did look at phosphorus in the zero tension lysimeters in that leachate. Phosphorus, we really don't expect to see any big difference between those systems at all. Phosphorus is not mobile. Nitrogen is very, very mobile and leaky and moves through the system and is hard to keep in place. Phosphorus, on the other hand, tends to get to one spot and it just stays there. And so unless you have extremely high levels of phosphorus in your soil and the soil can't bind onto the phosphorus anymore, you're going to expect to see that phosphorus is going to drop or not change or not move. And so that it's not really surprising. I'm not really sure why we're seeing this, although all these levels are very, very low. So it looks big here on this graph, but these are all very low levels of phosphorus. And so I'm not sure why we're seeing an increase in the mixture there. So bottom line, this is going to be a really short little talk for you. The average daily gains are higher in our grass legume mixtures when we add in our legumes, and that's great. I mean, that we can reduce our nitrogen fertilizer costs and we can improve our rates of gain. That's a win-win. Bottom line, that really helps the economic side of it. When we add in that legume, we are increasing the protein content of the forage. We expect then also to have higher nitrogen levels that are going through. So we're seeing increased nitrogen in the feces. We're seeing increased nitrogen in the urine, and we also saw increased nitrogen in the blood urea nitrogen levels. That's not unexpected. What we are pleasantly surprised with is that we're not seeing huge increases in nitrogen leaching when we actually get out into the field. The combination of those mixtures, having that legume and that grass, in many cases, we were actually lowering the amount of nitrate that was being leached. And even when we were seeing some increases, it was not tremendous. Those were not tremendous increases. Tall fescue, if you'll note, was always extremely low. And in every study I've worked with this tall fescue, it's a super great one at utilizing those nutrients. We really think that probably some of that difference, or that the reason we're not seeing huge increases in the nitrogen leaching in the field, is due to the rooting systems. When we can have two different types of rooting systems together, that can perhaps intercept those nutrients more efficiently. We now have root tube cages out there in the fields, and we are monitoring and taking scans of those roots to see if that can help explain what's going on, if it is the rooting system that's explaining it. The other part that could be playing a difference in this is that each species of plant has or supports its own different microbial populations. Different microbes like to pull in nitrogen and nutrients and tie them up, and they use them to grow and help break down organic matter and things in the soil. And then those microbes die and those nutrients are released, but they help tie them up so that they're not lost super quickly. Having two different sets of microbial populations may be able to better intercept those nutrients than having just one set of microbes that follow one species. So that's really kind of what we're seeing. We're seeing that we can add legumes into our system, improve our rates of gain, and we're not seeing negative consequences on the leaching side from the environmental side, and clay has not been seeing negative consequences on the reproductive side either. So with that, I will conclude my presentation.